Welcome to Biochemistry 414, Biochemical Techniques. Our model molecule today will be ribonuclease, SA, and we'll work on this throughout the whole semester. I'm your instructor for the class, Dr. Ayers. My administrative assistant is Mrs. Rausch. Our email addresses for both of us are listed there. The Canvas class website has contact information for us and for our teaching assistants. Office hours are posted on the website. Some of them will be by Zoom, and we'll use one link for everybody who is going, so you don't have to look for a link for each TA. You're welcome to come to anyone's office hours, or you can make an appointment. The primary goal of this semester will be to perform experiments as if you're in a real research lab. You will be using commercial protocols, research papers, protocols from other labs, not a cookbook manual. You will figure out how to do the experiments. Secondary goal of this class is writing. Most of the students will be writing intensive, 414W. If you ask your science professors how much time they're in the lab and how much time is spent doing writing, well, most of it's writing, research papers, grant proposals, reviewing these documents, reviewing the research papers of other scientists and other students, working on grant proposals of other scientists, dissertations, etc. And a lot of time is spent communicating in lab meetings, conferences, and teaching. Writing is a major portion of our time in science. Our molecule that we'll be using this semester is ribonuclease SA. It is one of the smallest enzymes known, only 96 amino acids, yet it manages to break down a very large substrate RNA. This molecule is highly acidic with a net charge of -7 at physiological pH. The substrate RNA also has a negative charge. So we have a negative enzyme and a negative substrate acting on each other. This is a great model system for studying protein folding. It helps us learn how to predict protein structure, and it has practical applications, everything from vaccines to laundry detergent. This semester, we will change a single amino acid to determine the effect on enzyme activity and stability. Here's an overview of the semester. We'll start off purifying plasmid DNA to work with, using restriction analysis to get an idea if it's the correct DNA. All of this on wild-type RNase. From there, we'll move on to site-directed mutagenesis. And now we will have two variants, a wild-type and a mutation E41K. From here, we'll go into inducing these proteins and overexpressing them, again, working on both of them. Purifying the proteins using osmotic shock, chromatography, concentrating the protein, using SDS-PAGE to analyze the steps of our protocol. And then we'll go on to enzyme kinetics to study the activity of the proteins and how altering a single base to alter a single amino acid to change charge might affect the activity. We'll also study protein folding to see the effect on there. We have a lot of electronic resources, a very detailed Canvas website. Grades are there, lectures, protocols, data that we need to share, worksheets, though we will give you a printed copy of each one, reference papers to use in your research, and we'll also use turnitin.com through the website. Note that all quizzes and exams are on computer using Lockdown Browser, so bring your laptop for this, although we will have a few computers available in the lab. The grade breakdown, all students will have worksheets that are worth 93 points, quizzes for 112 points, and there will be a quiz dropped. So if you're absent for one lab, you don't have to worry about that quiz. You get to drop one. We'll have a midterm and final exam. If you're in the writing intensive section, you will also be working on a paper in drafts throughout the semester. You'll have a midterm version, a final paper worth a total of 600 points, and peer review of each other's papers for 42 points. If you're absent without excuse, there are negative points on the final grade. And note that no grade changes will be made more than 1 week after posting. For the paper for 414W, it's one paper with drafts over the whole semester, so you don't have that frantic writing the whole thing at the very end. This breaks it down into smaller, more manageable pieces. It also helps you constantly review and understand what you've been doing in the lab. For peer review, you're graded on the peer review that you give to your fellow students, not how your peers review you. Note that 50% of the grade is from writing your paper over the semester. This is by university rules, which also state that you're not allowed to pass the class if you fail the writing portion. If you have an A on the whole rest of it, still doesn't matter. You have to get 70% or more on your paper to pass. Part of your grade will be lab performance. Much of the work that we do will be in pairs, but both partners should participate fully. If your partner does all of the work, you'll be graded accordingly. If you're a safety hazard, ignore rules, etc., your grade will reflect this. Note that on the final exam, a portion of it will be practical. You will be pipetting. You will be using specs. So if you have not been performing well throughout the semester, you probably won't do well on the final exam. Despite working in pairs, all of the work should be in your own words. You should submit your own graphs, not identical to your lab partner. If you answer questions the same, you'll get half credit. Throughout this semester, we'll be keeping lab notebooks. These are a record of what you did and why. They must be clear enough to be able to repeat the experiments in 5 years. Some experiments are complex and need notes. Sometimes you go into a research lab and you're told, get this notebook, repeat what they did and move on from there. If someone did not do a good notebook, you might have to repeat the work and figure it out. The same goes for your own experiments. Sometimes a company or a patent might be involved, and it might be the notebooks need to be dated and signed by a witness. For you, you'll need a bound composition notebook with lines for the next class, something like the marble covered notebook that you had in probably 5th grade, although you can have sparkles and puppy dogs or whatever on it. You cannot have a spiral notebook, no loose pages. The whole point is to be able to make sure that you show all your data and you're not hiding it. So you need a notebook where it would be obvious if you ripped pages out. Note that the quizzes, midterm, and final exam will all be open notebook. You're allowed to write whatever you want in there and use it for this. At the beginning of most classes, you'll have a quiz. This is to make sure you've prepared the experiments and you're not relying on your partner to get it all going. There'll be pre-lab questions, steps of the procedure, etc., and also some questions from previous labs to make sure you're keeping up with the material. For your notebook, put your name on it, your contact information. You do get to use this on the final exam. One semester, I had a student in late April. He had been dumpster diving. He was in the library, his notebook disappeared, and he thought it was in the dumpster. Luckily, somebody had picked it up and they called him and he got it in time for the final exam. Table of contents is optional. Make sure that you write legibly with ink. Again, you don't erase data, you don't hide it. So you must use ink, preferably on one side of the paper to make it easier to read. Before you come to class, you need to read the protocols and fill them out in ink, handwritten again to help you remember it and synthesize the information. Choose what you need to include, the date the experiment was performed, the purpose, 1 or 2 sentences with the overall reason for the experiment. Today I'm making a cell-free extract from bacteria containing our ribonuclease. Background, 1 or 2 paragraphs on what you're doing and why. Why do you care about ribonuclease? What are you using it for? Why are you making an extract? The details that you'll need to know. Then you'll also include a procedure that summarizes the protocols in enough detail to repeat them years later, even without any lab manual or protocols, published protocols. During class, you'll add your results and data analysis, raw data, tables, calculations, etc. Do note that worksheets are where you figure out most of your calculations. So perhaps you want that written in your notebook unless you're good at the calculations and don't need a sample to follow. You are allowed to glue in your graphs and pictures. Everything else must be handwritten though, to make sure you understand what's important. Sometimes there are no real results, perhaps only how you labeled your tubes. Move on to discussion, what your data means, discuss any problems. Did you get a higher delta G or a lower delta G? Did you have a problem getting your delta G? What does that mean? Conclusion 2, that must be supported by the data. Was your protein more active, your mutant, or less active than the wild type? What about stability? Your data has to support that. In the lab, we have workstations set up for each pair. The small drawer in front of you has safety glasses. There's a shared partner drawer between you and your partner, labeled your workstation number, for example, 1B, a very useful number to put on your samples. You often save them in the freezer for a month before you get around to using them all. Ditto if you're centrifuging samples in a shared centrifuge. It helps a lot to know what your sample is. If you just have your initials on it, it's harder to know who it goes to. In the drawer, you can see a lot of samples, a lot of Sharpies, tips, tubes, things like that, ruler. Note that the drawers are color-coded. This would be a yellow bench drawer. Racks are yellow or sometimes white if we don't have yellow for them. But several racks are yellow. The tubes are yellow. So again, when you have shared samples, you know you're looking for the yellow ones, not the blue or the green or the red. The pipettes that will be on your bench will also have yellow tape. So that will say it's the yellow bench. And there will be a roll of tape on the faucet for you to use for your samples. For each side of a bench, the central drawer has cuvettes in it for your use and extra tubes and tips when you use yours up. For safety in the lab, the safety points will be listed in the PowerPoints and on the whiteboard. There is no eating, drinking, or chewing gum in the lab. You must wear closed-toed shoes, have your hair tied back if it's long. We provide eye protection in the drawers in front of you. Also gloves in 5 sizes. There's a fire extinguisher at the front of the lab and a first aid kit. Note that the first aid kit really only has Band-Aids. We're not even allowed to keep aspirin because it expires. By each door into the lab, there's a big red button that is the emergency stop. If you need to turn off the gas and the electricity in an emergency, you push that and it'll turn them both off. In one of the sinks, there's an eye wash that we'll demonstrate in class. There are also eye washes in the hall on either side of the lab, as well as showers if you really have a problem with chemicals. There's a spill cut-up kit up front. Most of the time, paper towels are sufficient for our small spills. In the corner of the lab by the fume hood, there's a broken glass box, but let us know if you break anything. You do not put cuvettes or paper towels or anything in here, only glass. You can put your backpacks and purses on the hook under the bench or simply sling it under. If the backpacks are beside your Your chair, it's a safety hazard and we tend to trip over them, especially in case of a fire. By the door, there's a yellow binder with your materials safety data sheets for the chemicals that you work with. These are also on the computer. We've given you a copy of the safety agreement, so go ahead and sign that, documenting that you've been trained, and pass it to the TAs. In the lab, we have 3 types of waste containers. The first one is regular trash, several bins around the classroom. Most trash will go in there. There's a special glass trash box, which is labeled. Again, only glass, not plastic or cuvettes, etc. No paper towels. We have to pick that out before, if you put that in, and that does not make us happy. There's also biohazard bags. We'll set out a special container for biohazard DNA and bacteria that need to be autoclaved to be killed. This includes tubes that you've had, paper towels, etc. with them. On each bench, there's a small bag and an orange holder. Most days, when we're not working with biohazard, you can simply empty this into the big trash bins. Keeping the plastic bag, we try to limit the use of plastics. Once we start working with biohazardous material, we'll empty those into the large biohazard container and eventually remove the bag because it's been in contact with biohazards, and put out a fresh one for regular trash. Before you leave class each day, you should put everything away. Don't leave tips and tubes and stuff out. Put your pipettes back in the rack. Clean all of your dishes, giving them a rinse with distilled water in the 2 carboys we have in class, and place them on the dish cart upside down to dry. Empty the trash bags that are on the bench, keeping the bag again. Clean your bench with paper towels and cleanser, and report any missing items to the TA. Today, we're first going to work on your skills. A lot of people have been pipetting in research labs, and a lot of the time, they're very good at it. Some people don't really know how to pipette, and other people think they're good at pipetting and then find out they're not really good. Pipetting well is the one thing you can do best to get out of here quickly. If your experiments work the first time, then you can show us your data and leave. If not, we'll stay and make you stay till the whole 3 hours is up, repeating it until you get good data. We'll also work on spectrophotometry using Bradford assays. Next class, you'll start your lab notebook, and we'll start our ribonuclease project by purifying plasma DNA and doing restriction analysis to see what it looks like. When you have worksheets, you'll have a worksheet from today and then throughout. They're due at 10 p.m. on the schedule that is posted on the website. Today, we're going to use spectrophotometry and a Bradford assay to measure protein concentration. Once you purify your protein, you'll be doing this to determine the amount of protein you have at each step, how much you kept, how much you lost, etc. We're going to be using Kumasi Brilliant Blue, which binds to protein and changes the absorbance. If there's no protein, the maximum absorbance will be at 465 nanometers. Once the Kumasi binds to the protein, the maximum absorbance changes to 595 nanometers. We're going to follow that change in absorbance to determine how much protein there is in each solution. You're going to measure a series of protein dilutions, figure out the concentration in an unknown sample, following the instructions on worksheet 1. For our spectrophotometers, they're great specs. They hold 6 samples at once, 1 blank and 5 samples. We always use a blank in class. The light beam travels left to right, right to left. You can put the cuvettes in so that the 1 centimeter path length is through the light. You can see the image on the slide there. We're using semi-micro cuvettes to save reagents, so they're compressed at the bottom. If you put them in the right orientation, it'll be through 1 centimeter of liquid. If you have the wrong orientation, it'll be less than 1 centimeter. Our cuvettes are marked with an arrow at the top. You can match this with the arrow on the spec to make sure you're in the right direction. But basically, you're putting the light through the long dimension. When you put the blank in the spec in the first position, you'll do it like that. So the light goes side to side. And then the next sample and the next sample so that they rotate into the front position to be read in the correct orientation. Your absorbance should increase linearly with increasing concentration. If not, you need to redo any outliers. You should get a nice, straight, smooth graph like this. We have our data points that are not connected and that a trend line has been added. We have our equation of y equals 0.01x plus 0.002. Most of the time, you'll be forcing the graph through 0, and you will not have that point, the b, 0.002. If you're not getting close to 0 without forcing, then chances are you're not pipetting well. So this data is good. It goes close to 0. When we force it through 0, you'll probably have a y equals 0.01x with no b component. R squared is 0.998. The closer to 1, the better your data fits the line. Most of you should get an R squared about this high. If not, you're probably not pipetting well. So what happens if you have a point that's significantly above the line? Well, it's possible that you have not mixed. You really need to make sure you mix well throughout the class. Before you take a sample, once you make a reaction, once you have it in the cuvette, mixing will make your data better, and almost always it's very important to do. An easy fix for this is simply to mix it, remeasure it, and you'll probably find out it falls on the line. Sometimes you'll have a data point that's significantly lower than expected. In this case, you might have the cuvette in the wrong orientation. Again, quick, easy fix. Flip the cuvette, see if it now falls on the line. It's also possible that your reactions have not been made correctly. Some reactions take a long time to do, so make sure you pipette well, mix well, and put it in the correct orientation so you get the data the first time. Today, you're going to construct a standard curve with Excel using dilutions of known concentration to determine the concentration of an unknown solution. In this case, you'll be making a dilution series of BSA protein, bovine serum albumin, using a Bradford assay, which contains our Kumasi blue. You'll calculate the extinction coefficient for this, which is simply a proportionality constant that you use to determine the concentration of solution. From this, you'll determine the unknown concentration in your BSA sample that you don't know the concentration for. For your graph, make sure you put on a title, label the axes, add units. Usually you'll use scatter plot with the data points not connected. Add a trend line, equation, or squared. Again, that trend line should pass close to 0. Otherwise, you're not pipetting well. Some of the graphs will force through 0, some you can't. Graph title is one of the hardest things. Think of the type of graph, standard curve. It's pretty straightforward. You might be doing something like determining an extinction coefficient. Also, a Lineweaver-Burke plot. If you say Lineweaver-Burke, I know that there's an assay type, substrate, product, enzyme. You're measuring something in the spec. Positive y-intercept, negative x-intercept. And I care about Km and Vmax. All right, few slides left. What is wrong with this graph? Almost everything. The x-axis does not say what the alcohol is or the concentration units. The y-axis, you're measuring the absorbance. They're making it look like the units are nanometers, but you're setting your wavelength at a specific wavelength in measured in nanometers, not the absorbance. Absorbance does not have units. Title simply repeats the axes. That doesn't help at all. Series label on the right is useless. You can delete that. For their data, they connected the data points and added a trend line. You can see it makes it difficult to see the data. Do not connect the data points, simply add a trend line. You can see that they did graph their blank, which is the 0, 0, which they should. And also, their trend line does not go that close to 0, so they're probably not pipetting that well. Could this be the effect of drinking margaritas on a college student? You can't tell from the graph. You know, there's an increasing amount of alcohol and increasing absorbance. Here's a better version of the graph. We're following the conversion of ethanol to acetaldehyde, substrate and product, using the enzyme alcohol dehydrogenase. Now we know that we're working with ethanol on the x-axis, concentrations in millimolar. The y-axis, we're following the absorbance of NADH. You can't measure directly ethanol or acetaldehyde. It's linked to NADH, which we can measure at a wavelength of 340 nanometers. Here, the data points are not connected. Nice clean trend line. It's forced through 0. The equation is y equals 0.0123x. And the R squared is quite nice. How do we get our extinction coefficient from this? Beer's law, we need the extinction coefficient. So the extinction coefficient is simply the slope, 0.0123. How do we know the units? Well, let's take a look at our graph. Half length is 1 centimeter for us always in this class. It's not true of everything. Concentration, we know we're measuring in millimolar. Well, if units of absorbance, there are none for Beer's law, then that means that the other half of the other side of the equation, epsilon CL, the units have to cancel. You have a centimeter, you have a millimolar, so the extinction coefficient is the slope with the units of millimolar -1, centimeter -1. Finally, today, gloves are optional, eye protection is optional, cuvettes will go in the regular trash. The Bradford reagent, also called the Bio-Rad protein assay, does contain some phosphoric acid and methanol. It can cause burns if you dangle your fingers in it for a long time, and irreversible eye damage, but you're using small amounts. You're welcome to wear gloves or eye protection if you want to.